Firstly, I would like to explain about the process of photosynthesis. As we can see from the picture, sunlight, carbon dioxide, and water is required by the plant during photosynthesis, whereas oxygen and sugars such as glucose are released out during the process of photosynthesis. This is the chemical equation of the photosynthesis. 6 molecules of carbon dioxide plus 6 molecules of water will form 1 molecule of glucose and 6 molecules of oxygen, and photosynthesis requires chlorophyll and sunlight. Photosynthesis occurs in 2 stages, that is, light-dependent reaction and light-independent reaction. Next, I would like to demonstrate about light-dependent reaction. Light-dependent reaction can also be called light reaction. This is a simple picture about how light-dependent reaction occurs. It occurs in thylakoid membrane and it consists of 2 types, that is cyclic photophosphorylation and non-cyclic photophosphorylation. It is about non-cyclic photophosphorylation. This is a simple picture about the non-cyclic photophosphorylation, and the explanation will be on the next page. Light energy is absorbed by accessory pigments of photosystem II, or we can call it as PS2. Light energy is transferred from one accessory pigment molecule to another molecule. It is transferred to the reaction center of pigment P680 in PS2. P680 becomes photoactivated. An electron excites to a higher energy level in the reaction center. The electron is captured by the primary electron acceptor, that is the phosphatine. Photolysis of water occurs. Photolysis of water is a process of an enzyme called Z protein that contains manganese, extracts electrons from water and supplies to P680, replacing the electrons that the chlorophyll molecule lost. And a water molecule splits into 2 hydrogen ions and an oxygen atom, which immediately combines with another oxygen atom to form O2. Each photoexcited electron passes from the primary electron acceptor from PS2 to photosystem 1 or PS1. Electrons passes via an electron transport chain. Chloroplast version consists of an electron carrier called plastoquinone, a complex of 2 cytochromes, and a copper-containing protein called plastocyanin. As electrons cascade down the chain, the exergonic fall to a lower energy level is harnessed by the thylakoid membrane to produce ATP. When an electron reaches the bottom of the electron transport chain, it fills the electron hole in P700. Primary electron acceptor of PS1 passes the photoexcited electrons to ferredoxin. An enzyme called NADP+ reductase transfers the electrons from ferredoxin to NADP+. NADP+ reductase adds a proton to form NADPH. NADPH is released into the stroma. Proton gradient created in the thylakoid space is used in chemiosmosis. ATP is produced from ADP plus inorganic phosphate. Next, I would like to explain about the cyclic photophosphorylation. This is a simple picture about the process of cyclic photophosphorylation. The yellow arrow refers to the cyclic photophosphorylation. Firstly, light energy is absorbed by accessory molecules and is transferred to P700 in photosystem 1. Electron from P700 is photoexcited and passed on to an electron acceptor called iron sulfur. Iron sulfur then passes the electron to ferredoxin. In cyclic photophosphorylation, an alternative route is used. The electron is passed from ferredoxin to cytochrome B6F complex. Energy is released during the electron flow, and this is used in chemiosmosis to generate more ATP. The electron is then transferred to the plastocyanin. Electron is transferred back to photosystem 1, and it can then be photoactivated again.
Going to talk about light-independent reactions. For the questions, what is the light-independent reactions and where is it occur? As we know, the light-independent reaction is the reactions that do not require the light energy, and the main part of the light-independent reaction is the Krebs cycle. So the Krebs cycle is going to occur in the stroma of the chloroplast, and Krebs cycle is an enzyme control process. So for each turn of the Krebs cycle, 1 carbon is used and is used to make a useful product, which is sugar. So let's learn more details about the Krebs cycles. There are 3 main phases of the Krebs cycles: phase 1, phase 2, and phase 3. So the phase 1 is known as carbonylation, whereas the phase 2 is known as reductions, and the last phase 3 is known as regeneration of carbon dioxide acceptors or regenerations of IUPP. Let's focus on our carbon cycle diagram. So from the carbon cycle diagrams, we can see that our carbon cycle will start with a 5-carbon compound called ribulose bisphosphate, which is sometimes abbreviated to IUPP. So in the phase 1 carbon reactions, the IUPP will fix with the atmospheric carbon dioxide to form a 6-carbon unstable substance, which finally it will produce 2 molecules of the 3-phosphoglycerate for each carbon dioxide molecules. And this reaction is catalyzed by the enzyme called Rubisco. While in the phase 2 reductions, the hydrolysis of the ATP will occur and the 3-phosphoglycerate is phosphorylated to form 1,3-bisphosphoglycerate. Next, the NADPH from the light-dependent reactions will oxidize back to NADP+. So the 1,3-bisphosphoglycerate is dephosphorylated, which means that the phosphate group is removed and it will reduce to form glyceraldehyde 3-phosphate. While in the last phase, phase 3, 1/6 of the G3P is used to produce a sugar, whereas the 5/6 of the G3P is used to regenerate the IUPP to allow the cycle to continue. So this process will require the ATP. So the IUPP is now prepared to fix with the carbon dioxide again. So the carbon cycle will repeat. We are moved to the photosynthesis summary. Here's the overview of how the two sides of the photosynthesis, that the light-dependent reactions and the light-independent reactions, interact. So here's our chloroplast, and we can see on our left-hand side we got the thylakoid arranged into the start of the grana, and that is where the light-dependent reactions occur. So as the waters and the light energies come in, the light reactions will occur, and it will produce the waste product, which is oxygen. So at the same time, it also produces the usefully ATP and NADPH. So the ATP and NADPH are going to other parts of the photosynthesis, which is carbon cycle in the stroma. So for this, the carbon dioxide, ATP, and NADPH is needed. Finally, the carbon cycle will produce 1/6 of the sugars, NADP+, and ADP.
We are going to make an experiment on photosynthesis. So here are the tools and materials that we'll need. So we have table lamp, water, baking soda, teaspoon, syringe, transparent glass, dish soap, spinach leaves, and single hole puncher. To start the experiment, first we are going to make carbon dioxide solution. So we are going to pour water into the glass, and then we are going to put a drop of dish soap, then we are going to put a little bit of baking soda, and mix it gently. And then we are going to use hole puncher to cut 10 leaf discs from the spinach leaf. Make sure the size of the leaf discs are almost in the same size. And now I'm going to place the disc into a syringe. So put it into the syringe gently. And I'm going to tap the syringe so that they do not stick on the side of the syringe. And then I'm going to push the plunger back into the syringe until a small volume of air is left. So until this much. And then I'm going to suck up several milliliters of baking soda solution into the syringe. So the leaf discs float on the water because there are air from the air pockets within the leaf that makes them float. And then push out the air from the syringe. And I'm going to put my finger on the opening of the syringe, and I'm going to pull the plunger to create a vacuum. And hold the vacuum for 10 to 15 seconds while shaking the syringe slightly. I'm going to release my finger and release the vacuum, and then now you can see that all the leaf discs has sink to the bottom. I'm going to pour the leaf discs into the solution. To place the glass under the lamp and observe the leaf until they float onto the water surface.
The photosynthesis process will utilize the light energy, water, and carbon dioxide to synthesize the energy in the form of sugar, while at the same time, the oxygen gas will be released as the product of this reaction. Those are the conclusions about the photosynthesis process. Thank you for paying attention about our presentation.